All right, ladies and gentlemen, welcome back. So, in order to be able to talk about how plants actually harness light energy and convert it into sugars via photosynthesis, we have to take a step back and understand how light actually works. So, your chloroplasts really are these solar powered chemical factories. We're going to look at those thylakoid membranes and the chlorophyll that is trapped within them, and they're going to take in that light energy through these light reactions, convert it into the chemical energy of ATP, NADPH, so that the Calvin cycle can ultimately produce those sugars. But when we look at the sunlight that comes in, we need a little bit of a physics lesson here. So there are only certain types of light that those plants are actually capable of harnessing and using. So what we're looking at here is that light is in the form of electromagnetic energy or electromagnetic radiation. And that light energy travels in waves, right? So think of waves in the ocean. Okay, and the distance from one peak to the next is what we talk about as a wavelength, and it can be crest to crest, so peak to peak, or trough to trough. It's kind of the same basic idea. Some of those wavelengths are really far apart, and so the distance between those two peaks is really long, and sometimes they come really close together, and so the peaks are really short. The wavelengths are really short. So that's what determines the type of electromagnetic energy you're dealing with. How far apart are the troughs or how close are they? And so when I look at gamma rays, right, I'm looking at 10 to the negative fifth nanometers. That means that that wavelength is super, super, super tiny. And so the troughs come really, really close together. And as a result, the frequency is really high. On the opposite end of the spectrum is 10 to the third meters. So things like radio waves have huge, huge wavelengths, and so the distance between their peaks is really, really long. And so when we look at sort of the penetrating power, if you will, gamma rays have a lot of energy, come really short bursts, but they can't travel really far. Wavelengths, on the other hand, are very long distances, not only between the peaks, but also in terms of how far they can travel. But they don't have a whole lot of energy behind them. So the shorter the wavelength, the closer together they are, the more energy they have. Longer wavelengths, further apart they are, but the less energy that they contain behind them. So light is an interesting thing. So when we look at the way light acts, and if you want the details on this, go talk to a physicist. This is not my area. But the basic idea I get is that light can act as waves and have energy, but they also can act as discrete particles called photons. And when we're looking at the context of how light works within photosynthesis, it's going to kind of act like both. And so I like to think of those photons as sort of ice cream flavors, right? The different photons correspond to the different wavelengths of energy that are coming behind them, all right? So when we look at this electromagnetic spectrum, all of this energy is coming um, in different forms. When we look at sunlight specifically, it's going to dip into this UV radiation component, right? That's why we wear sunscreen, because that can be potentially damaging because it's higher energy, shorter wavelengths. Goes all the way through the visible light, which is what's being split out here so you can see it, as well as into the infrared, which is what we basically perceive as heat. There are only certain wavelengths of that energy through that UV, through the visible, and into the infrared. The plants aren't designed to be able to harness all of that. Instead, they can only harness components of it. Only certain wavelengths of that energy that the sun is bringing in are actually capable of driving photosynthesis. So go back to that photon idea. Only certain photons are going to be captured by that chlorophyll. So only certain flavors, if you will, of light are usable by the plant. So what do the plants use then, and how do we know? So let's answer this question. Why are plants green? Well, here's the thing. I've been throwing this word chlorophyll around, right? It is a major plant pigment, and pigments are substances that absorb visible light. Okay? So when that sunlight comes in and hits a pigment, regardless of whether it's chlorophyll or pigments in a piece of paper that's been printed, or pigments in your clothes, or pigments in your own skin, that wavelength comes in all those visible colors, right? The entire rainbow of colors is captured within that white light that we see coming from the sun, or for that matter, even from the lights in the room. And when that light hits an object that is pigmented, some of that energy gets absorbed. Some of that light, some of those colors gets absorbed and stay with the pigment itself. Other wavelengths of light get transmitted, meaning they pass straight through, and or they get reflected. And typically the reflected and the transmitted light wavelengths are usually the same colors. Okay, so what gets absorbed stays here. What gets reflected and what gets transmitted is what gets bounced back to your eyes. So the colors that get absorbed are the colors you don't see when you look at an object. You're seeing color, you're perceiving color as the white light being scattered and what that object isn't keeping. Okay, and so that reflected light gets bounced back up to your eye, and then you've got these things called cones, so light sensitive cells in your eyeball that are capable of detecting different colors depending on which of them get stimulated, right? So you actually have red, green, and blue cones, all right, and it's very similar if you've ever walked up to an old school TV and been able to see the pixels, the different colors, right? You step back and you can see a huge range, but it's a matter of that electronic device highlighting and intensifying certain colors and certain ratios within that very small little pixel so that when you step back, you can see that dynamic range of color. Your eyes are essentially doing the same thing, but it's electrical signals that it's sending to your brain, and your brain has to interpret it. So for people who are colorblind, they have some sort of mutation or uh, retinal damage that doesn't allow one or all of those cones to work and function properly to send the signals to your brain so your brain can't perceive certain components of color. Pretty cool. All right. So when we look at plants, when we look at green leaves, they are green to us because that chlorophyll is taking in all of those colors except for the green. And we're actually going to see it's green and yellow, but if you've ever mixed green and yellow paint together, it still looks pretty green, and so you're not really distinguishing the difference there. So the green and yellow light is what's getting transmitted and reflected. That's coming back to your eyes. That's what's stimulating those cones in your eyes to tell your brain, hey, this is green. All right. When you look at something that's white, all of those colors are being transmitted and reflected at once. When you're looking at something black, it's absorbing everything, which is pretty cool. So how do we measure photosynthetic pigments? How do we know that this is actually what's happening beyond just understanding the physics of pigments? Well, we can put them through a spectrophotometer, which is what you're looking at here. And spectrophotometers are these devices that measure the pigment's ability to absorb various wavelengths. Um, so it's a machine. You take a solution, in this case they're showing you chlorophyll here, and you stick it into the machine, and that machine has a white light and a refractive prism so we can break out and adjust the wavelengths that are going through this slit one at a time. We transmit that light through our specimen, through our solution, and there's a photoelectric tube on the other side that is measuring the absorption. Okay, depending on the spectrophotometer you have, you can adjust parameters and get it to measure absorption versus transmittance. so on and so forth. And really, absorption is the exact opposite of transmission. So if you know what one is, then you know what the opposite is. Um, so when we put through that chlorophyll and we shine that green light through it, all of that energy is hitting that photoelectric tube, measuring it as a really high transmittance, really low absorption as a result. And so we know that that solution, in this case chlorophyll, isn't holding on to any of that green light. I change the color to something like blue, and now I'm getting a very little amount of energy, very little amount of light that is hitting that photoelectric tube, really low transmittance, and as a result, a really high absorption. So it's hanging on to that. So we can use this to decipher different pigments' roles in a plant. Because keep in mind, not every single plant is green, not every component of a plant is green. So what pigments are there? What things are they absorbing versus transmitting? And then we can start backtracking and trying to figure out, well, why is it doing that? What is its function? What is its purpose? And how is it doing it? So what we can do is create these absorption versus action spectra. And this is a pretty common one, just FYI, that tends to show up on exams. So let's spend a little bit of time here. Because what we look at in terms of an absorption spectrum versus an action spectrum are very similar, but not identical. So an absorption spectra is something that we might create via one of those spectrophotometers, right? And it's just the graph plotting the light's absorption versus its wavelength. So shining that light of different wavelengths, so purple, blue, green, yellow, orange, red, through that pigment and seeing what gets absorbed versus what gets transmitted. Okay. The action spectrum, on the other hand, is a pretty cool experiment that was done by this gentleman named Engelmann. And what he looked at is here's a filament of algae, right? And he was able to basically prism out the rainbow from white light and look at that algae and where it was doing photosynthesis as opposed to the collection of aerobic bacteria. Where did they grow best? And what he figured out is that this action spectrum followed very similarly to the absorption spectrum. Okay. So those aerobic bacteria want to use as much oxygen as possible to do their cellular respiration process, and so they're going to congregate where that oxygen is being produced the most, and he was able to overlay the colors of the spectra to see where the photosynthetic rate increased based on that, and use those aerobic bacteria as an indicator for the rate of rate of photosynthesis that was occurring. So here's just the basics, right? He did this all the way back in 1883, which I think is kind of clever because they definitely had to have a basic understanding of the way light worked, and they did at this point. But to be able to use those bacteria, know what they were doing, know that photosynthesis, at least in some component, was occurring, and be able to piece all of this together is really pretty cool. So here's the thing. While I'm on this topic, I said that these action spectra and these absorption spectra don't line up perfectly, right? And the reason for that is because your plants don't have just a single pigment. Most plants will have two different forms of chlorophyll A and chlorophyll B, as well as these accessory carotenoids. And this allows the plant to sort of broaden out the amount of energy that it's taking in and using for photosynthesis. So if I look at chlorophyll A, it is very much just purple and into the red. Chlorophyll B now dips into the blue, but you don't get a whole lot. You get a little bit down into the orange, but not much. All right, so not quite as high. And then the carotenoids are just up here in this purple and blue. There's nothing down here at all. So when we look at the rates of photosynthesis versus those wavelengths, again, they don't match perfectly, and that's because of those accessory pigments. So here's what chlorophyll looks like. All right, the only difference between the A and the B form, interestingly enough, is that the A has a CH3 here, chlorophyll B has a CHO. Crazy how just that little bit of structural difference means some difference here in terms of what they are capable of absorbing, which tells you that that particular functional group there does mean something. All right. And then we also have those carotenoids, and those carotenoids help to absorb some additional light energy at that blue and purple range, right? That's the high energy side of things, without allowing that high energy to do too much damage to the chlorophyll molecules, which are going to be kind of the workhorses here. Um, but interestingly enough, think about when you would see those carotenoids. If you look, right, they absorb in that purple and blue, but can't absorb anything down here. So when do you see red, orange, yellow leaves? In the fall. That chlorophyll actually breaks down earlier in the season as the sunlight starts to become less during the day, less intense, less duration, the whole 9 yards. That chlorophyll starts to break down. Those carotenoids stay on just a little bit longer to hang on to just a little bit more of that energy to continue photosynthesis for a little bit longer into the season, so that that plant can store that energy in its tissues, whether it's just its roots, stems, whatever the case might be, until winter officially hits so that it can overwinter itself. It's got enough energy to do some really low-level metabolic processes all winter long, until it sprouts new leaves and has the energy to sprout those new leaves because of the energy that it's stored. Not only do those carotenoids allow to extend the wavelengths that it can absorb during the spring and summer, protect the chlorophyll during that time, but also then extends the season, if you will, for it to be able to absorb energy for just a little bit longer into the fall, which is pretty cool. So how do we know all of this is happening and what is it doing? Well, we can look at how chlorophyll actually uses that light and be able to harness it to trap it. So anytime that a pigment absorbs light, the atoms within that pigment go from its ground state to its excited state. So what does that mean? Let's back this up a step. So remember that your electrons in an atom are generally hanging out in those orbitals, right? 2 electrons in the first, 8 in the second, 8 in the third, and then you start getting into subshells. But those electrons hang out in the orbitals that are associated with the energy that they have. So if they have more energy, they're at a higher orbital. If they have lower energy, they are at a lower orbital closer to the nucleus. Well, when that light comes in, here's where that photon component comes in. When that light comes in as a photon, it's going to hit those electrons and push its energy into them. And what that means then is that those electrons bump up. They've been given more energy, and as a result, they bump up to a higher orbital. But that's not stable. All right, so this excited state, these electrons that have been given extra energy aren't stable, and so eventually they'll fall back down to their ground state. They'll lose that energy and go back to where they originally were located in the atom's structure. When that happens, when that excited electron falls back down to ground state, we lose some of that energy as heat, and then the rest of that energy has to go somewhere. When we look at your light reactions, that's going to be really important to see where that energy goes. But if I don't give it anywhere to go, it'll actually come out as fluorescence. It'll glow. And so if you isolate chlorophyll molecules from a plant, don't allow it to be part of that chloroplast structure anymore. You just have pure chlorophyll, and you shine a UV light up to it, it actually glows red, which is pretty cool. So you're exciting those electrons, and as they are dropping back down to their ground state continuously, you're getting this transmission of reddish fluorescence out of the chlorophyll. It's pretty neat. Your plants, though, are going to take that energy and capture it, and that's where those light reactions are going to come really into play. Just sort of an analogy to help you kind of think about what we're talking about here. I always like to think of those electrons as kindergartners, right? And if I come into a kindergarten classroom and I just throw candy and cookies and cake and all sorts of sugar at those kids, what are they going to do? They're going to end up on a sugar high, right? They're not going to be able to stay in their seats anymore, and they're going to be bouncing off the walls. And that's kind of what these electrons are. They're bouncing off the walls of their orbitals and popping up higher orbitals as a result, jumping on the tables, if you will, because they're excited. No kindergartner can stay in that state if that constant flux of sugar isn't there, or in this case, that constant flux of light isn't there. So eventually they fall back down to the ground state. They end up zonked out on the floor, and that energy has gone somewhere. They've burned it all off. In this case, it comes off as heat and fluorescence.